Pyrogene and Endotoxin Testing, Questions and Answers, the new U.S. FDA guidance for industry. This guidance document was developed by the U.S. Department of Health and Human Services Food and Drug Administration, Center for Drug Evaluation and Research, CDER, Center for Biologics Evaluation and Research, CBER, Center for Veterinary Medicine, CVM, Center for Devices and Radiological Health, CDRH, and the Office of Regulatory Affairs, ORA. This document was made official in June 2012. If you'd like an additional copy of this document, you can get it from the address shown on the screen. You can also get it on the web by following the address seen below. Introduction. The FDA guidance provides recommendations for biological products, drugs, and device manufacturers on the FDA's current thinking concerning the testing and acceptance criteria in USP Chapter 85 and USP Chapter 161 and the Association for the Advancement of Medical Instrumentation, or AAMI, Standard S72 2002, Revision 2010. The FDA guidance is not intended to cover the entire subject of pyrogen and endotoxin testing. It addresses those current issues that may be subject to misinterpretation and are not covered in compendial procedures or in currently available guidance documents. Background. The FDA has accepted the use of the LAL test in lieu of the rabbit pyrogen test for well over 30 years. The conditions for using the LAL test were first published in the Federal Register in 1977, and the citation is 42 Federal Register 57749. Development of the guidance document for LAL testing began in 1983, and the guidance for the validation of the Limulus amoebocyte lysate test as an end product test for human and animal parenteral drugs, biological products, and medical devices was published in 1987. So why was the 1987 guidance document withdrawn? Well, frankly, it was obsolete and no longer reflected FDA's current thinking regarding LAL testing. The 1987 guidance, by the time it was withdrawn, was actually over 25 years old. And the FDA believes that the information published in the United States Pharmacopoeia Chapter 85 BET and the AAMI guidance documents provide sufficient information for industry in order to do this test. Guidance documents. FDA guidance documents are just that, guidance. The information provided in guidance documents is not binding to FDA or to industry. The new FDA LAL guidance document has changed format from the previous guidance document. This document is not nearly as prescriptive as the 1987 document. This document is in the form of a question and answer document regarding regulatory issues only, and it defers technical issues to the United States Pharmacopoeia, ISO, AAMI, and other documents. While the information provided in guidance documents is not binding to FDA or to industry, firms should consult with their reviewer if they decide not to follow the recommendations found in the guidance document. The topics discussed in the FDA Q&A document, which was released in June of 2012, include sampling, retesting, sample handling, sample pooling, alternative assays, switching alternative methods, Appendix E, the endotoxin limit tables, establishing limits, quality by design, safety threshold versus process control, USP 151, veterinary drugs, establishing limits for multiple species, medical device limits, therapeutic screening and assay dilutions, and also the use of a controlled standard endotoxin. Sampling procedures. How do I establish a sampling plan for in-process testing and finished product release? The document states that firms should consider the potential for contamination in raw materials, in-process materials, and the finished product. Also, that firms should take into account impact of an in-process hold time, endotoxin removal steps, and finished product. Regarding the sampling procedures, The FDA says that firms should consider their sampling plan dynamic. They should start with a maximum coverage, and they should adjust their sampling plan as they gain confidence in the prevention of endotoxins in their manufacturing process. Firms can no longer rely on the guidance document to prescribe the number of samples that must be tested, as was in the previous document. The sampling plan that the firm derives must be justified and must be presented to the FDA in an application such as an IND, NDA, BLA, or other such documents. The next question is regarding retesting and when is retesting appropriate. The guidance document states that when conflicting results occur within a test run, that the user should consult USP Chapter 85 gel clot limit tests interpretation for guidance on repeat testing. As per Chapter 85, if the test failure occurred at less than the maximum valid dilution, the test should be repeated using a greater dilution, not exceeding the maximum valid dilution. The user should record the failure of the lab results, and all testing procedures should be specified in advance in written SOPs approved by the firm's quality unit. The next question is regarding sampling, storage, and handling, and that is sampling, storage, and handling important? The guidance document says yes, the ability to detect endotoxin can be affected by storage and handling. Firms should establish procedures for storing and handling, and then parenthetically, which includes product mixing, samples for endotoxin analysis using laboratory data that demonstrates the stability of the assayable endotoxin content. The document goes on to state that procedures should now always be validated to assure that endotoxin content is not lost during storage, and that if frozen storage is required, consideration must be given to the effects of freeze-thaw on endotoxin recovery. The previous thought was that samples for endotoxin testing could be stored at 2 to 8 degrees for up to 24 hours before testing, and that the sample should be frozen if the sample could not be tested within 24 hours. The FDA has changed their opinion of that practice since the original document. Sample storage procedures should now always be validated to assure that endotoxin content is not lost during storage. This can be accomplished by spiking a sample with a known quantity of endotoxin and assay that sample at defined intervals after storage at that temperature. For example, a sample of a drug product could be spiked at 1 EU per mil and placed at 2 to 8 degrees C. The sample would then be assayed every 2 or 3 hours to show that the endotoxin content did not drop off. If frozen storage is required, the consideration must be given for the effects of freeze-thaw on endotoxin recovery. For example, a sample would be inoculated at 1 EU per mil, vortexed, and then aliquoted into separate vials, which are then frozen. At specific time intervals, an aliquot would be thawed and assayed, and this would continue for the length of time the firm wished to store the sample frozen. The next question was regarding pooling of samples. Can finished product samples for analysis of bacterial endotoxin be pooled into a composite sample prior to analysis? And the guidance document says yes. Pooling would generally be accepted for small volume parenterals of 100 milliliters or less, as long as the maximum valid dilution is adjusted to a proportional lower value because of the potential for diluting a unit containing harmful levels of endotoxin with other units containing lower endotoxin levels. FDA suggested in this example that if you pool 3 samples, then you divide your maximum valid dilution by 3. The document states that pools should be aliquots made up after 30 seconds of vigorous mixing, from each of the product containers, and that the individual containers should be retained for possible retesting in the event of an out of specification result. Finished medical devices may also be pooled into a composite sample and assayed, and that testing for medical devices should be conducted using the rinsing, eluting, and sampling techniques described in ISO 10993-1 and ISO 10993-12. The guidance document goes on to state that sampling may be adjusted for special situations. The pool must be maintained at the appropriate conditions to promote stability, and some product types should not be pooled. Examples of such would be products with an initial low maximum valid dilution, products manufactured as a suspension, and in-process samples from different in-process stages. The appropriate storage conditions must be determined on a product-by-product basis, as was discussed under sample storage. For example, for an initial low maximum valid dilution, if pooling 3 samples and the MVD is 1 to 3, you might be testing right at the MVD. Products manufactured as a suspension may have sample homogeneity issues which may present significant interference issues, and it would be extremely difficult to ensure the homogeneity of the samples. The next question is about alternative assays. May a firm use alternative assays to those in the USP for a compendial article? And the answer to that from the guidance document is yes. Firms may use alternative methods and/or procedures if they provide advantages in terms of accuracy, selectivity, or adaptability to automation or computerized data reduction, and in other special circumstances. Such alternative tests should be validated as described in the USP General Chapter 1225, Validation of Compendial Methods, and be shown to achieve equivalent or better results. Better validation should be in accordance with the requirements of USP 85 and of USP Chapter 1225. The guidance document goes on to state that recombinant horseshoe crab factor C assays may also be used. An example of such is Pyrogene. Bruce Loverock, Barry Simon, Alan Baines, and Allen Burgenson have already published the validation of 10 different representative pharmaceutical products in the January-February 2010 issue of Pharmacopoeia Forum, the official journal of the United States Pharmacopoeia. Our data shows that Pyrogene works as good as or better than classical LAL methods when testing these products. Our validation went through the validation categories found in USP Chapter 1225. If you have not seen this article, a copy is available free of charge from the online Pharmacopoeia Forum of the United States Pharmacopoeia. There is a one-time registration fee to get access to this free online reference document. The next question is regarding transitioning from one alternative BET method to another. And the question is, what is the best process for transitioning from one alternative BET method to another? This is done by comparing the two tests to verify the equivalence of the new method using USP Chapter 1225. For drug, animal drug, and biological products, the transition to a new method should be submitted in a prior approval supplement, or PAS, or by using a comparability protocol, CP, before using the new method for release. For medical devices, a 30-day notice may be appropriate for changes to quality control testing for all BET testing. This addresses changing from one alternative test, for example, recombinant factor C, to the MAT test, to another, but does not mean changing from one compendial method to another. However, if going from a compendial method to an alternative assay, the firm must first perform the validation and then submit to FDA in the form of a pre-approval submission. Once a firm develops a history with the alternative test, they may then submit a comparability protocol. The CP is simply an outline of what testing will be done, What parameters must be met each time before the product will be routinely tested with the alternative test. Once the CP is approved, the product submission for each product after that is reduced from a PAS to a CBE 30, or changes being effected in 30 days. A CBE 30 allows the firm to release the product into interstate commerce 30 days after the FDA receives the submission and not have to wait until FDA approves the submission. This could result in the time saving of several months per product. The next question is regarding Appendix E of the 1987 guidance document. And the question is, what happened to the endotoxin limit table in Appendix E of the 1987 guidance? The endotoxin limit table is out of date due to increases in the number of dosages and drug strengths since publication of the 1987 guidance document. Therefore, the customer or drug sponsor should use the calculation methods provided in the USP or AMI documents. Monograph limits may not account for current product strengths or dosage regimes, and endotoxin limits should be checked using the calculations recommended in the standards. The FDA will no longer allow submissions to contain references to the endotoxin limits found in Appendix E. The FDA wants to see the actual calculated limits based on K/M, including how the calculation was done for each product. You must show the math in the submission. Limits for multi-component products should be calculated based on the expected contribution of endotoxin from each component. For example, with pre-filled glass syringes, the contribution of the syringe must also be taken into account. For products that would be mixed prior to administration, the content of the mixed product may not be able to be tested. Therefore, the content would be determined by adding the content of each of the components. The sum of the components must be below the limit of the firm established for the combined product. Intrathecal product limit specifications have not changed. Ophthalmic products are a new category in endotoxin testing due to endotoxin issues causing health issues after being administered during intraocular surgery. The next area addressed in the guidance document is quality by design concepts and endotoxin limits. The document states that strategy should be based upon product and process understanding in combination with risk management for consistent final product quality. It states that in-process testing should be used to evaluate the production process areas at risk of endotoxin formation or incursion. Furthermore, finished product release specifications should be considered when determining in-process limits for each phase of manufacturing. And in quotes, qualitative testing may be preferable to limit testing to facilitate product quality trending and to identify and correct excursions before they exceed the specifications and cause product failure. These are the same ideas regarding in-process testing and raw material testing that have been promoted for over 20 years, and actually much longer. The firm should assess the quality of their incoming raw materials and assess the relative contribution of each raw material in the final product. Animal or plant materials may have greater contribution of endotoxin than a synthetic inorganic material. The raw materials would be trended over time to assess their suppliers' quality systems. Trending also allows manufacturers to not use a raw material if it exceeded the limit set for that raw material to prevent the final finished product from failing the release assay. This is just standard process control. The next area that the guidance document discusses is the rabbit pyrogen test. And the question is, when is the USP Chapter 151 pyrogenicity test, or the rabbit test, appropriate? The document states that for certain biological products, 21 CFR 610.13b requires a rabbit pyrogen test. However, this test may be waived if a method equal to the rabbit pyrogen test is demonstrated in accordance to 21 CFR 610.9. For human and animal drugs, some USP monographs still require a rabbit pyrogen test. The user may substitute an endotoxin test or alternative cell-based assay if the firm can demonstrate equivalent pyrogen detection. Testing is performed in a side-by-side comparison of the rapid pyrogen test at various concentrations of the test article versus testing of the same dilutions using LAL or an alternative test such as Lonza's Pyrogene. The rapid pyrogen test may also be required if the presence of non-endotoxin pyrogens could be present, an example of which would be cytokines or TNF or the like, or gram-positive pyrogens, or when the bacterial endotoxins test is subject to interferences that cannot be overcome. An example would be if you have to dilute your sample past the maximum valid dilution for the LAL test. One thing that must be remembered is not all pyrogens are endotoxin. This must be assessed during preclinical development, especially for cell-based therapies and biologics, where the cells used in the manufacturing process may release chemical signals due to stresses on the production system. Additionally, other microbes may release pyrogenic substances into the product if contaminated. These substances may not be as potent as endotoxin, but will still cause a pyrogenic reaction if found in the product. The next area for discussion in the guidance document are limits for veterinary products that targets multiple species. And the question is, how would an appropriate endotoxin limit be determined for a veterinary product that targets multiple species? And the document states that for use in multiple species, the limit should be based on the maximum product dose used on the smallest species. For use on juvenile and adult animals, the juvenile is usually considered the worst case. And if the weight of the animal is required to calculate the dose, you should use an average weight for that species. And an example of where those weights could be found is in the waivers of in vivo demonstration of bioequivalence of animal drugs, insoluble powder, oral dosage form products, and type A medicated articles. The next area for discussion is limits for medical devices, and the user should refer to USP Chapter 85 and USP 161 for information. The user should also refer to the FDA guidance for industry document for full clarification. Limits for devices are found in USP Chapter 161, transfusion and infusion assemblies, and similar medical devices. The endotoxin limit depends on the route of administration, intended use of the device, and what the device contacts, for example, blood or the cardiovascular system, lymphatic system, cerebrospinal fluid, intrathecal routes of administration, permanently implanted devices, and devices implanted subcutaneously. The FDA added information regarding flushing the device for catheters where the internal surface is labeled as non-pyrogenic, disassembly or cutting the device into pieces for devices that are too large to be adequately rinsed, and also immersion for implantable devices. For some examples of limits of medical devices, we find that the device limit stayed the same as which was listed in the AMI guidance document, and that's 20 EUs per device. And an example would be a device that contacts the cardiovascular system or lymphatic system. And the extraction volume would be 40 milliliters of non-pyrogenic water, such as water for injection or LAL reagent water, and testing 10 devices. And the concentration limit would be, in this example, 0.5 EUs per ml. If the device contacted cerebral spinal fluid, the limit would be 2.15 EUs per device. And again, using the 40 ml extraction volume, the limit would be 0.06 EUs per ml. Intraocular devices is a new category stemming from the use of solutions which were not originally meant for use internally during eye surgery, which caused problems attributed to endotoxin contamination. The new guidance states that a lower limit may apply, but the firm could contact the appropriate review division for specific recommendations. This means that the limits will be evaluated by reviewers on a case-by-case basis, and a rote formula may not be appropriate. The next area of discussion is screening for therapeutic drug products, and the question is, what is the FDA's expectation for regular screening of therapeutic drug products? The document states that the undiluted product should be screened, and that if ingredients interfere with the LAL test, the user should dilute to overcome the interference, and that the calculated MVD is the dilution of the sample at which the endotoxin limit would be detected, but it should not be the regular testing dilution. If interference is encountered during the product development, the firm should determine the lowest product dilution that would neutralize the interfering condition, and this is known as the minimal inhibitory concentration, or MIC. Finally, the guidance document goes on to discuss controlled standard endotoxins and their use, and the question is, are controlled standard endotoxins, or CSEs, still acceptable for use in running BT assays? Controlled standard endotoxins are endotoxin preparations traceable in their calibration to international reference endotoxin standards, such as those found in the United States Pharmacopoeia, or EP. They're manufactured and certified by an LAL manufacturer for use with a specific lot of reagent under defined assay conditions. CSEs have become an accepted source for preparation of standard curve calibrators and assay controls. They have provided cost savings to LAL users and help preserve the inventory of primary standards. Furthermore, the FDA encourages the continued use of controlled standard endotoxins. Finally, I'd like to thank you for your attention during this presentation, and if you have any questions, you can contact me at the information you see on your screen, or you can also contact your sales representative or Lonza Scientific Support. Thank you very much.